much about glycans, so I'll make a small introduction. So the protein you see revolving in the middle, that's the S protein of the SARS-CoV-2 virus, and everything hairy you see on the surface are the glycans. So we cannot understand the function of this protein if we are ignoring the glycans. So the best analogy I can make is this bird on the left, where you have a bird without a single feather. This bird is not a functional bird, and protein without its glycans is just like a bird without its feathers. It cannot perform all functions, because proteins, glycans can change the structure of a protein. If you change the structure of a glycans attached to immunoglobulin, it will convert it from pro-inflammatory to anti-inflammatory molecule. So if you are studying only polypeptide part, and we use this bird as analogy, if you try to understand flight, and you use a birdless, featherless bird as a model, this will be the closest to the flight you can come. Just bird walking around, because bird without its feathers cannot fly. Protein without its glycans cannot perform full functions. Glycans give the full complexity of a protein structure. They explain protein function. And just a word of caution at the beginning, this has nothing to do with the glycation. Glycation is a random damage. It's a chemical reaction between glucose and the amino acids. We quantify it as HbA1c, advanced glycation end products. This is something completely different. Glycation, random damage. Glycosylation, sophisticated modification, which makes proteins way more elaborate. Actually, glycosylation adds additional layer of complexity between gene sequence and a protein function. So polypeptide part, this is so bacteria. You have a gene, you have a protein, you have a function. This is the way how bacteria function. And E. coli has 5,000 genes. We have 20,000 genes. We are not only 5 times more complicated than an E. coli. So at one point in evolution, when we were becoming multicellular, we invented glycosylation. We invented a network interactions of dozens of proteins which work together to make a glycan structure, which then becomes a part of a protein. An alternative glycosylation, attaching different glycans to the same glycosylation site, is analogous to coding mutation. It's changing the protein function, but it's being inherited as a complex trait. And this is something we really clearly demonstrated when we looked at the study of cohorts of over 100 mice strains generated as a collaborative cross cohort. So they started from the 8 founder strains, crossed them for 3 generations, and then inbred the cousins into strains. And some of these cousins, some of these strains became structurally and functionally distinct from the rest of the population without a single mutation. They became different just because they reshuffled alleles in this complex network which glycosylates proteins. And we see the same in humans. We are different. On the right-hand side of this panel, it's just 6 random individuals, different color, different glycan, IgG glycan composition. People are different. Big part of inter-individual variation, so what makes us different from another human, are glycans. Actually, blood groups are also glycans. We just don't think it in that way. So glycans are the molecules which can help us understand why are people different. So why people don't look glycans more? Why didn't you learn about glycans during your studies? And the answer is because glycans are extremely complicated. You can have many different structures attached to the same glycosylation site. You can have multiple glycosylation sites on the same protein, and it really takes many chemical steps to analyze a glycan, especially in a high-throughput manner.